Hi everyone, my name is Roxane Legaie. I am a senior bioinformatician at Monash Health. I have worked for many years in the research sector and I recently moved to Melbourne in order to bring my career to the clinical side of things. Firstly, I would like to give you a brief introduction to bioinformatics, which is a field of science that combines computer science and statistics for the analysis and interpretation of large-scale biological data, such as DNA or RNA sequences, in order to identify genes or mutations of interest. Clinical applications will depend on the effect of the mutations found across patients. If they are responsible for a particular disease, we can use this information to provide a diagnostic or develop novel therapies. And if they are providing resistance to treatment, we can do personalized medicine through targeted therapy. And all those applications together have created what we call clinical genomics through state-of-the-art bioinformatics. For the next part of this talk, I would like to take you through some of the steps of a standard bioinformatics pipeline for clinical genomics. During variant identification, it is important to differentiate germline and somatic mutations. Germline mutations are present in the whole body of the patient, while somatic mutations are only present in a tumor site, for example. Which is why, to identify cancer-related mutations, we need to also collect a control sample from the blood or saliva. And the downstream analysis is also quite different. Once the variants have been identified, a curator needs to filter them based on population frequency and predicted consequences at the transcript and protein level. It is also important to keep in mind that a variant that was found to be non-pathogenic or of unknown effect today might be classified as pathogenic in the future, in which case a clinical record needs to be updated and the corresponding patients informed. Variance classification and reporting is what I call the Pandora box. There is lots of ethics around genomics and incidental finding. For example, if a patient is tested for cardiac disease genes and the analysis finds high-risk mutations in the BRCA genes, do we tell the patient that they might develop breast cancer later? Do they want to know? And what if their life insurance gets access to this data? Also, genomics data is what we call big data. We are talking gigs of data per patient. And that makes it very difficult to understand, analyze, and visualize. There are many tools for data interpretation, but most of them are not very intuitive. Also, storing and sharing variance data worldwide is crucial to the development of our knowledge and understanding of genomics information. To conclude, clinical genomics is already here. It's not a thing of the future anymore. However, there are still many challenges that need to be addressed, in particular the need for automated processes and worldwide data sharing through international databases and local initiatives, such as the Melbourne Genomics Health Alliance and the Human Biome Project. And finally, I truly believe that clinical genomics could be much faster and more cost-effective if we had some sort of bedside device that could run the end-to-end process on the spot. And this is now possible thanks to the recent single-molecule sequencing technologies that can run a sequencing experiment in a USB stick-like device and produce and analyze genomics data within a couple of hours only. So watch this space. Thank you.